I'm delighted to have the opportunity to discuss with you oceans, climate, and human health, especially to be able to take what we have learned from cholera to apply in predicting risk of COVID-19. The work that we have done has focused predominantly on water-related diseases, namely cholera, a diarrheal disease, which afflicts about 1.5 billion to 2 billion cases each year, with at least 1 million to 2 million deaths. It is a continuing pandemic. As a matter of fact, it's an acute water-related diarrheal disease, and we are in the seventh pandemic of cholera, which started in the 1960s. Cholera cases are recorded as pandemics. Now, cholera occurs in about 50 countries. It affects about 7 million people. And the Bengal Delta, unfairly, is known as the native homeland of cholera. But in fact, up until 100 years ago, when we instituted chlorination and safe distribution of water, we had cholera in the US and in Europe in strong epidemic cases. Now, the important point to make is that the early work we began in the 1960s in the Chesapeake Bay, and these are the stations that were sampled on a regular basis and even continue to be sampled today. That allowed us to make some rather important discoveries about cholera, particularly the fact that it's a naturally occurring bacterium in the aquatic environment and that it's associated with copepods. In other words, the vector of cholera is the copepod. The copepod, of course, has a chitin exoskeleton. And interestingly, the Vibrio has a powerful chitinase, which breaks down the chitin of the exoskeleton. In addition, it has a powerful proteolytic enzyme, which allows it to break the case of the gravid female so that the eggs can be sent into the water column during maturation. One of the important studies that we have developed from the early work is a model um, which shows, in fact, that in the spring months, when the temperature of the water is warm and there's plenty of sunlight, the phytoplankton become dominant, they bloom. The zooplankton feed on the phytoplankton, and then when the nutrient source is depleted, the crash of the zooplankton releases the Vibrio cholerae into the water column, and then transmission occurs via drinking water that's not been purified or treated. And then fecal shedding, person to person transmission, can take over, and that has been the component of cholera that has been most intensely studied. We were criticized when we first proposed this model 25 years ago that it probably didn't work in Bangladesh. So the challenge was to go to Bangladesh, which we did, working in the Sundarbans in MATLAB, one of the remote located laboratories in Bangladesh that is now an international laboratory of research. And indeed, the observations that we made showed that the water is in fact being taken by the village women, collected for drinking water in the household in the calashes, and then carried to the house for the day for the water supply. But also, it's a source of washing utensils for bathing, etc. In the far corner is the latrine, so draining into the pond. So there is this constant intermixing, hence the difficulty in maintaining conditions that would prevent cholera. What we know is that with poor conditions, there's person to person transmission, but really the source of the bacteria is the environment. And that was a novel idea, and I would say not so readily accepted. It took about 20 years for the fact that it's an aquatic bacterium to reach the textbooks. Now, what's also interesting is that the relationship of chlorophyll um, uh, and water temperature as well as air temperature playing a role, led us to the hypothesis that we could use Landsat, which was launched in 1985, and that allowed us to be able to measure um, the sea surface temperature as well as chlorophyll and sea surface height. And we figured that we could use these data and the sensor to allow us to predict the risk of cholera. And sure enough, we found that the red line are the cases of cholera in the Sundarbans of Bangladesh, and then the blue line is the water temperature just offshore. This was pretty crude, but it was a remarkable correlation, and this was published in the Proceedings of the National Academy of Sciences 20 years ago. We subsequently carried out a study of Hurricane Matthew, and we were able to predict where in Haiti the risk of cholera was highest. And in fact, that turned out to be the accurate prediction. And we subsequently, when analyzed Yemen, when a massive epidemic occurred in Yemen in 2017, we showed where the high risk area was, and in fact, the actual cases were where we predicted. We were asked by the British Aid Agency to work with them and with UNICEF to provide on a monthly basis a 4 week predictive capacity for where the highest risk for cholera would be in that country, which would allow then the British Aid Agency and UNICEF to locate physicians, supplies, medical supplies, and safe water. This led to a significant reduction in cholera in Yemen in 2018, and we have continued to provide the data every month with a 4 week advance prediction of risk of cholera. Now, what this has to do with COVID turns out to be quite interesting. COVID, of course, as we now know in the very rapid accumulation of information from December and January of this year to the present, is that the disease, the virus, affects the lungs, the liver, the kidneys, the intestines, and even the brain, because in the most severe cases, there's a loss of smell and a loss of taste. Now, this is a virus, and I've been describing Vibrio cholerae, which is a bacterium. Nevertheless, what we were able to do with our very sensitive DNA sequencing techniques was to identify the SARS-CoV-2 virus and its variants in stool. Now, what has been determined is that um, this is a slide showing 41 different patients, and the red line, horizontal, shows the time of a positive throat swab test, and the yellow-orange shows the persistent shedding of the virus even when the test goes negative with the patient recovering. So the virus continues to be shed in the stool. Um, this allowed us then to use this information as we had done for detecting cholera to measure the virus presence and number in different wastewater stations, sewage treatment plants around Maryland. We are now in the process of doing a 3 year study with the Department of Environment, Department of Health, and the Governor's Office in Maryland, which gives us an, an estimate of the viruses present. In this case, it's results from one site, Frederick, Maryland, and in July there was a big peak. Now the two lines are two different sets of genes that we've used to track the virus. The blue is the end target gene set that the Center for Disease Control recommends, and the orange line represents another set of genes, the CRF lab target gene that we use in addition, and that gives us very strong um, um, reliance of the report that we can then give the governor. And indeed, this peak in the sewage sample numbers was followed as was predicted within 4 to 7 days, with cases appearing in the community. So this provides advance warning, but also when the numbers go way down, it means that distancing, masking, hand washing, and refraining from meeting in large numbers or groups is working. And then the economy can be opened. So it's new information for the governor. But what's really fascinating is the fact that we were able to take the model from the cholera work and modify it for COVID-19 risk. And on the left is our risk map for April, May in one of our very early applications. And this was the actual case distribution in April, May in the United States. A remarkably good fit, so that what we have learned about cholera has allowed us then for this model to be applied for predicting cases of COVID-19. I will close by saying that when we do science, we need to remember that we must help those who are in need from the science. And so knowing that copepods carry the vibrios and particulate matter are what the vibrio is attached to, we figured that if we could remove the copepods and the particulate matter, we could reduce cholera. And in a 3-year study with several hundred villages involved and several thousands of people, we were able to do a study and show that when sari cloth was folded 4 or 5 times, it gave a 20 micrometer mesh filter. And copepods are about 200 to 300 micrometers. So they were trapped. And we knew from our lab tests that this folded sari cloth could work effectively to remove about 99% of the bacteria. And also for the women, we were able to show that this clean water after filtration was much better and safer for them than the turbid water with things swimming in it. So we were able to reduce cholera by 50%. Now, nylon mesh had been used in Africa, but it's very expensive. It had been used for dracunculiasis, which turns out to have an intermediate stage in copepods. But the sari cloth was very effective and very inexpensive, almost nothing, because used sari cloth is available in every village. So I'll close by thanking my collaborators at the International Center for Diarrheal Diseases Research in Bangladesh, at the Calcutta Cholera Hospital, and students at the University of Maryland. In addition, colleagues from other agencies, specifically NASA, but NIAID as well, and from other countries and other universities in the US. And then finally, the work is written up in very great detail in the book that was published last August by Simon and Schuster. So I thank you for the ability to present this information and hope you will find it useful and interesting.